Third-generation nanopore sequencing is a powerful novel sequencing technology that allows to very easily obtain sequence information from a wide variety of samples. The main advantages of this technique are the portability of the sequencing device, the extremely long read length, and the real-time availability of data. Nanopore sequencing is a particularly helpful technology during disease outbreaks in remote locations. It has successfully been used in field laboratories during and after Ebola virus outbreaks in West and Central Africa.
While nanopore sequencing and the use of the MinION device for this purpose is rather easy, care must be taken during library preparation and during flow cell loading.
To begin this procedure, set up a touchdown PCR to amplify the cDNA with primer set 1 using a hot start high-fidelity DNA polymerase with the appropriate reaction buffer in a 50 microliter reaction volume with 1 microliter template. If possible, set up the reaction on ice or in a cool block at 4 degrees Celsius. Incubate the reaction in a thermocycler as outlined in the text protocol. After this, transfer 50 microliters of the PCR product into a 1.5 milliliter DNA low-binding reaction tube. Resuspend the magnetic beads thoroughly by vortexing. Then, add 50 microliters of beads to the PCR reaction and mix well. Incubate the sample on a rotating mixer for 5 minutes at room temperature while rotating at 15 rpm.